So, Sam, you've watched Finding Nemo, right?
Yeah, of course. I've cried many times watching Finding Nemo, which I know was not actually your question.
That's okay. I do feel like you answered it in the best way. So you remember that Nemo and his dad Marlin lived in this funny little house. It looked like a pink bowl with wavy French fries sticking out of it.
Yes.
Yeah, I feel like I can just see it. And that house was actually an animal. More specifically, it was a sea anemone.
So it's in a group of animals called cnidarians. So those are stinging-celled animals. And the big group of cnidarians includes corals, like on a coral reef, jellyfish, box jellies.
That's Anne Tarrant, a senior scientist at Woods Hole Oceanographic Institution, where her lab has set up a system with an infrared camera to watch sea anemones move around.
And so what we see with those animals is they're more active at night. So they're little night owl anemones. They crawl around at night and they're more sluggish during the day.
And Anne is watching those anemones to answer a really fundamental but complicated question. How do they know what time it is? Welcome to Tiny Matters, a science podcast about the little things that have a big impact on our society, past and present. I'm Deboki Chakravarti, and I'm joined by my co-host, Sam Jones. Sam, we're in my least favorite time of the year. Daylight savings has ended, things get super dark at 5 p.m., and everything just feels wrong.
Yeah, I also can't stand it. I go to pick my baby up from daycare at 5 and it's already dark. I feel like I'm ready for bed at 4:30 p.m.
Absolutely.
Yeah.
We're recording this at like almost 3 p.m. and I'm like, after this, straight to bed.
Yeah, time to put on my jammies. And, beyond that, a big part of why things feel weird with daylight savings is because it messes up our circadian clock, which isn't a physical timepiece.
They're clocks that are inside of an organism, and it's a way that an organism can have an internal sense of what time it is.
We all probably have our routines over the course of the day. And of course, we probably all need to look at a watch every so often just to make sure we're sticking to the schedule. But our underlying biological needs that shape those routines are driven by the way our body keeps track of the 24-hour cycle.
For example, waking, sleeping, eating. During certain time of the day, we crave for coffee. Certain time of day, we feel hungry if there's no food supplied.
That's Hua Lu, a professor in the Department of Biological Sciences at the University of Maryland, Baltimore County.
The circadian clock is widespread. Many organisms, including photosynthetic bacteria, fungi, animals, and plants, all have the circadian system and the ability to keep time.
Hua studies the circadian clock in plants, and descriptions of how plants follow some kind of daily rhythm go a long way back. In the 4th century BCE, a scribe to Alexander the Great described the leaves of Tamarindus indica, which would open during the day and close at night. But circadian clocks don't just track the time of day. They can also give organisms a sense of longer periods of time.
At the seasonal level, plants know when to flower and when to set seeds. And at the seasonal level, some animals know when to get food for the winter, when to hibernate, and when to migrate.
When I think of an actual physical clock, I think of all sorts of dials and gears that have to be in motion to keep track of time.
Yeah.
And our circadian clock is kind of like that, except that the dials and gears are made up of environmental signals and the molecules inside of our cells.
So the clocks are what we call entrained. They get synchronized by signals from the environment.
That's Anne again. These signals from the environment are called Zeitgebers, which is German for time givers, which is a very cool name.
It is.
So the most common one we think about is light. So if you're in and out of light and dark over the course of a day, that'll help line up your clock with everybody else's clock. But then in theory, if you were kept in a dark room all day, maybe working at your computer, your internal clock would keep running. And so that's a way that all the different processes inside an organism, all their cells, all their tissues, basically know what time it is.
Anne told us that plants can even use different wavelengths of light to entrain their clocks to incorporate more information about what their environment looks like. And alongside light, there are other signals like temperature and food.
We're usually eating during the day, or animals are eating whenever they're active and awake. And so the food coming into their body can act as a really powerful entraining signal.
That's really interesting. You're talking about food as sort of an input, but to me, naively, I would think of food as also being shaped by these circadian rhythms. I think of breakfast time as more a function of my circadian clock rather than something that would be training it. I'm curious, how do you think about that?
I think that's one of those things where we want things to be synchronized in the body. If we're sort of adapted to being awake during the day when it's light out and warm out and we're eating, I think all those signals go together. So I agree with you absolutely that appetite, is something that we think of as being governed by the clock to some extent. But then again, that food itself entrains the clock. So that's one that kind of reinforces itself.
So there are these Zeitgebers, like food and light, that are coming in from the environment, and organisms need to have a way to sense and process that information. For light, a common class of proteins in our cells that do this are called cryptochromes, in Greek meaning hidden color.
And these cryptochromes are present in all different kinds of animals. They're present in flies, they're present in butterflies, they're present in worms, but they're also present in plants. And what we think is that that was an independent evolution where these cryptochrome molecules are really good at detecting blue light. They do other things too, but they're really good at detecting blue light. And so they get incorporated into these circadian clocks. So that's something that shows up over and over is different organisms figuring out ways to detect light and then input it into the clock.
And once these Zeitgebers are sensed by the organism, they'll activate a pair of proteins called clock and cycle.
Very cute.
I know, this is a total side note, but the circadian clock people have a lot of great protein names. There's a timeless gene and a clockwork orange gene.
So fun.
I know, it's so much fun. And we can't get into what they all do, but they're all working together to create what's called the central oscillator, which produces a signal that will go up during the day and then down at night. So basically, the central oscillator is like a wave that cycles over a 24-hour period, which helps the cells in an organism keep track of time. And this overall structure is pretty universal for organisms that have circadian rhythms.
So although the core clock components or the genes making up the central oscillator of plants differ greatly from those of animals, the basic mechanisms of action of different central oscillators is highly conserved in plants and animals.
Despite these similarities, Hua told us that there are still some key differences in how clocks can be implemented in different organisms. For example, with animals, there's generally a central clock in the brain, which then connects to peripheral clocks located in the organs.
The central clock perceives light cues and synchronizes the peripheral clocks through neuron signals, body temperature regulation, hormones, and feeding patterns to ensure body function alignment with environmental cues. Plants, on the other hand, have relatively similar clock systems that are decentralized.
That means that if you look at a tree, the leaves are running their own little clocks to sense their environment and keep track of time, and they coordinate with other leaves close by to keep things synchronized.
So one of the things I've always been really stuck on when I've read about circadian rhythms is how do you know that there's actually a circadian clock shaping an organism's behavior? So for example, going back to breakfast, how do I know that the reason I want breakfast in the morning is because my body is tuned to want food at certain times of the day, and not just because I see sunshine and decide that that means I should have food?
Duboki, really loves breakfast, everyone.
Apparently, it's the only thing on my schedule I can really commit to. So anyway, I asked Anne how her lab studies this in their model organism of choice, the starlit sea anemone, and she told us that they keep sea anemones in a cycle of light and dark over a 24-hour period, just like they would experience in nature. They'll measure whatever behavior they're looking at and watch it follow that 24-hour cycle, and then they turn the lights off.
And for it to really be circadian, the cycle should keep going on this 24-hour period, even though it's fully dark.
That makes sense. So if it isn't circadian, you won't see whatever behavior continuing in the same way.
Exactly. Like if the anemone mostly moves at night and stays still during the day, then they should continue to follow that routine over a 24-hour period, even if you keep things dark. And this is actually pretty important to test for another reason.
A lot of marine animals do have circadian rhythms, but some of them have circadian rhythms. So you asked a question about Zeitgebers, or external signals, and one that I didn't mention. Another signal that marine animals are exposed to are the tides. So the tides, depending on where you are in the ocean, they're often twice a day. And so some marine animals have rhythms that are about 12.4 hours rather than 24-hour rhythms.
That's pretty wild.
I know. As I said before, Anne studies the starlit sea anemone, which is also known as Nematostella victensis, and her lab confirmed that they were on a circadian clock, not a circadian one. And her lab was also curious about something else. How do circadian clocks handle two signals from the environment, especially if they seem to be in conflict with each other?
Usually, we think about when we're exposed to natural sensory cues, they're what we're sort of adapted for. They're all lined up and synchronized so that our bodies can work efficiently. And so if we think of a human, we're used to it being light during the day, dark at night, or eating during the day. So that would be if the cues are all aligned. That's what the organism sort of expects. That's probably what the cells expect to see in terms of input. But if we think about a human, if you're maybe up late because you have to work a night job, but you're exposed to light during the day when you go outside a little bit, and then you're, up all night, and maybe you're eating at unusual times, those cues are off cycle from what the body might be adapted to.
When you have cues that seem to be off from each other, you get what's called sensory conflict. I know that I have experienced this a number of times, most recently when managing the absurdity that is newborn sleep. But other animals can potentially deal with sensory conflict too. For example, with some cnidarians, scientists are worried about the impact light pollution might have on them.
And that's something people have been concerned about for corals. The idea that, if you think about the ancient world, there weren't people in cities. And so the animals really evolved for it to be fully dark at night, except for, moon and starlight. And as our societies have developed, we've had more and more light at night. And so that causes a perhaps sensory conflict to a variety of marine organisms.
And while Anne told us that the starlit sea anemone has so far appeared to be robust to light pollution, there's still helpful organisms to study how animals deal with sensory conflict. More specifically, Anne's lab looked at the impact of light and temperature on the movement of Nematostella. Remember, these are little night owls, so they're supposed to move around at night. And light and temperature are cues that should be linked together. Usually as the sun comes up, the day gets brighter and warmer. And as we get to nighttime, things get darker and cooler.
And so I had a wonderful graduate student in my lab, Corey Berger, who worked on this project. The idea was looking at what happens when we put a Nematostella in an environment where we could independently manipulate light and temperature.
So Corey started by lining the cues up on the same schedule, and then he began to spread them apart, first by 2 hours, then 4 hours, then 6, then 8. And he looked at how that offset affected when Nematostella would be moving around.
And what he found, it was surprising to, I think, both of us, was that Nematostella was very tolerant of that. And they maintained a 24-hour rhythm pretty strongly until the light and temperature were offset by like 10 hours.
They found that when the cues were just slightly offset from each other, the Nematostella seemed to primarily follow the temperature cycle to keep their rhythm going. But that only lasted up until a certain point.
When they were fully offset, fully opposite to each other, that's when we saw no rhythm. They completely lost their rhythmicity. It's possible that those two signals, when they're really, really offset, they're telling the clock different things, and that it's causing processes to work opposite to each other in a way that they sort of cancel out.
Yeah, and I'm curious too, this is sort of premised on the idea that there are multiple inputs to the circadian clock. And I'm wondering what the advantage of that would be for organisms, because it seems like in some cases it's creating confusion. So is there an advantage to having multiple inputs?
So there could be natural times when you don't always have the cue that you're used to. And what we think is that having multiple inputs provides a way to reinforce the clock and provide stability to it. So even if maybe you're not seeing the light very well because you're burrowed in the ground or something, you still have a temperature signal. And so the redundancy, we think, might be important.
Given how important circadian clocks are to regulating reactions throughout our bodies, it makes sense that you would want a little redundancy. Anne told us that even really small-scale processes can be affected by circadian rhythms, like when our cells divide or when DNA gets repaired. One theory for why we have circadian rhythms is that it's a way to keep the cells in our bodies synchronized. But for something like controlling cell division, it might also be protecting our DNA.
When we think about nature, a lot of organisms are exposed to ultraviolet radiation. And so, especially if you were a very small single-celled organism, that ultraviolet radiation can be really damaging. It damages DNA. That's why we get skin cancers, for example. And your cells have ways of repairing a lot of DNA damage. And so you might want to think about when your DNA is being repaired, when it's dividing, in the context of whether or not there's ultraviolet radiation around. So that's thought to be one of the really fundamental drivers of some parts of circadian rhythm.
And Hua told us another important reason for circadian clocks.
Resources are limited for all organisms. This is true for humans. It's also true for other animals and the plants.
The idea of figuring out how to use those limited resources is really important to Hua's work studying how plants defend themselves against threats like bacteria and other pathogens. One of the important things to realize is that plants don't have the same resources and advantages as us when it comes to infections.
Plants do not have specialized immune cells that can move around to detect and fight against the pathogens. Neither do they have adaptive immunity that involves T cells and B cells and antibodies to help fight against the infections. Plants do not move around, obviously, right? Plant cells do not move either inside of the plant body. Plants only use innate immunity to defend against the pathogens.
Innate immunity means that those are the built-in defenses that have evolved to recognize certain threats, and they include both physical and chemical barriers. These defenses are pretty similar to how our own bodies work. For example, we have skin to keep out pathogens.
Plants, like us, they have a surface epidermal cells. And then they also have surface structures, such as trichomes. These are hair-like structure on their body, and a layer of waxes provide protection.
And like us, plants also have chemical compounds that can fight against threats, alongside a surveillance system made up of different molecules on their cells that recognize pathogens and mount a low level of defense against them.
Microbes have also evolved their own mechanisms to counter these defenses, like producing molecules called effector proteins.
These effector proteins can use various kinds of chemical mechanisms to compromise the host immunity and to cause disease.
And of course, this is all just one long battle that's playing out over eons, which means that plants have evolved their own response in the form of resistance proteins.
They could use so-called resistance proteins to specifically recognize some effector proteins from the pathogen. and subsequently activate a much stronger form of defense. This so-called much stronger form of defense typically is associated with a suicidal response.
Killing off a part of itself helps the plant limit the spread of the pathogen. But as cool as these defenses are, they all still require resources.
Too much defense is not good. Too little is not good. Activate defense at the wrong time is not good. We need to activate defense at the right time and, of course, at the right place.
And you know what's great at keeping time? Yes, the circadian clock.
Of course.
Scientists have found that plants are able to time their defenses against pathogens and pests using their circadian clock to basically best anticipate when those attacks might come. For example, Hua told us about a study involving a species of moth called cabbage loopers that likes to munch on the leaves of mustard plants, particularly around noon. Those plants in turn can make compounds to become unappealing to their predators. The thale cress plant, also called Arabidopsis, makes a defense hormone called jasmonic acid over a 24-hour cycle. The hormone peaks at noon when the cabbage looper likes to feed. So the scientists decided to try something really cool.
They switched the day-night for the insect, which made the insect to have a peak eating behavior at the opposite time to the peak of jasmonic acid accumulation. This phenomenon is called out of phase. The insect gained twice as much weight compared to when the insect eating behavior and the jasmonic acid accumulation are in phase.
I think this is super neat. Plants basically have a little timer that helps them keep track of their local pests so that their chemical defenses are all set up in time.
Yeah, it is really cool. And the circadian clock also plays an important role in setting up physical defenses for the plant. The plant surface is covered in little pores called stomata, whose opening and closing are controlled by the circadian clock.
Normally, stomata open in the day and close at night. This process is important for photosynthesis and water exchange.
If you're a plant, you want your stomata to be open, but that opening is also a door for pathogens. In Hua's lab, they decided to see how plants coordinate the timing of these physical structures with pathogen defense. So they started by spraying the bacteria Pseudomonas syringae on the leaves of an Arabidopsis plant.
We found that there's a lot more bacterial growth in the plants when the infection was conducted in the morning than when the infection was done at night. This makes sense, right? Because in the morning, stomata open, allowing the bacteria to get inside of the leaf tissue relatively easily. At night, stomata close, therefore restrict passage infection.
So that showed that the closed stomata are important for defending against bacterial infections at night. And next, they wanted to see what would happen if Pseudomonas bypassed the stomata and got inside the leaf tissue. So they loaded up a small syringe with the bacteria and injected it into the leaf.
Surprisingly, we find that with this infection method, there was a lot more bacterial growth in the plants when the infection was conducted at night than when the infection was done in the morning.
So Hua's lab had basically found that there were different defense strategies being employed by the plant, and that these strategies were tied to their circadian rhythm. During the night, the plants can rely on their closed stomata to keep out intruders. But during the day, when the stomata are open, the plant turns to other defense mechanisms for protection. And these different strategies are carefully timed so that the plant isn't wasting precious resources.
And this is just one example of how circadian clocks can shape organisms. But there are so many more, not just for plants, but for us. And that can have huge impacts on our health.
We're awake during the day, but there's people, of course, who are awake at night for a variety of reasons. So it could be parents with young children. A really common one is people who work a night shift. And there's been studies where shift workers experience some metabolic diseases where they don't metabolize food exactly the same way as people who work on a day shift. So they're more likely to have diabetes-like diseases.
So that's not exactly great news for everyone dealing with weird work schedules or babies or jet lag or daylight savings. But at least from a science perspective, I think it's really cool that if you look all around you, you'll see plants and animals and other organisms that are keeping track of time even when they don't know it. They're taking in so many signals and creating schedules for their biological routines without knowing it. And we're all just keeping time together.
That is really cool.
It is. But also, I'm still tired.
Yeah, me too.
Well, on that note, tiny show and tell time?
It is tiny show and tell time. I can go. Oh, look at us.
Yeah, we're just so excited.
Yeah.
I'm also really curious if we did the same one today.
I don't know. I kind of doubt it, but also, I don't know. I did an evolution one.
Me
and
too.
I know you're, oh no, I know you're big on evolution.
And I did one that I was like, I think Sam would enjoy this one. Okay, I will go first.
Okay.
Sam, this is about dogs.
Oh, I will. Okay, we did not do the same one, and I do love this one. Please continue.
So, dogs today are super diverse, and people have generally attributed that to us and all of the breeding that we've done over the last few centuries. I didn't realize this, but like we've probably domesticated dogs for like a very long time, obviously, right? Like the oldest estimate is like 33,000 years ago. Ancient DNA studies say it's probably more like 11,000 years ago.
Yeah.
But even though we like domesticated them a long time ago, we didn't really start doing these committed breeding programs until the middle of the 19th century.
Yeah. Which is so I just like, that is mind blowing. Like,
right?
Usually we're talking in millions of years, hundreds of thousands of years.
Yeah.
And even tens of thousands of years is like fast.
Yeah.
Hundreds of years is unbelievable.
Absolutely. It's funny because I think you're pointing out how funny it is that like we've like created like basically all of these little dog species like over a very short time span. It's also wild to me that it kind of took us that long to get to that point, like that we've been domesticating dogs for so long.
Right.
But we weren't like,
I want this squishy faced dog who does this thing. I think, well, just because the utility changed so much, right? Like it goes from having a dog that it looks a lot like a wolf is a form of protection. Having my dog that is just a really squishy thing is just cute and fun and sweet and there for my emotional support.
I want to see your dog like herd a sheep.
Oh,
just one sheep.
No. They'd like run the other way. I'm like, I don't know what that is. I gotta go.
So basically, like, we've really attributed the diversity of dogs today to that fact. But like maybe we're wrong. So there's a recent study by scientists in France who are looking at dog and wolf skulls going back 50,000 years ago. And of those skulls, the one that was oldest, most definitively a dog skull, went back 11,000 years ago, which is interesting in part because it lines up with that ancient DNA study.
Right.
But the thing that really captured their interest is the fact that the diversity in size and shape of the dog skulls was pretty like big. Like there was more diversity there than they expected.
11,000 years ago?
Yeah, like of these old dog skulls. They didn't have quite as extreme of features like things you would see today, like, the squishy faces and stuff. They didn't get as much of a hint of those. But there's just more diversity than they expected. And so that might mean that like dogs got their diversity even before they, were being bred by us. And so it might have been things like climate and geography that shaped, which makes sense in some ways, right?
Totally.
I think it's just more about like understanding the degree of like just how much our breeding has shaped what they look like today, or the diversity of them today versus maybe there's a lot of diversity there already to begin with.
Yeah. Yeah, no, that is really interesting. And it's just also, wild. I know you said before you were like all these different species of dog, but I know you meant like types of dog because
I was trying to think of the right word, but
breeds, but like it is crazy that they're all the same species.
I know.
Is my point. Like this makes no sense. I laugh about that where I'm like, if a wolf saw my dog, they would laugh and maybe want to eat them. It's a little complicated. But so that's really interesting. I never actually thought about the role that environment beyond like human intervention
could have
on the development of, different dog breeds.
Right.
Whoa.
But it makes sense. I mean, it makes sense. Like, we did our cat evolution episode, and there's a lot of interesting stuff there about how similar the wild cat is, like the most recent, like, basically ancestor of the cat is to like the domesticated cat. And I feel like, I don't know, pets are so weird. Like, they are. Yeah. It's not just pets, it's animals in general. Like, it's not just because there are pets that it's weird. But there's an element of it becoming weird because they're so entwined with our history and the way that we've interacted with animals that it's just fascinating.
Yeah, absolutely.
Well, I'm glad that we didn't do the same one.
I know. I got a little bit nervous there because I know that you've been really on an evolution kick. Yeah. Yeah. Absolutely. Maybe it's not a kick anymore. It's just a new fascination.
I know. Yeah. Yeah. This is really turning into like our little pet corners of what we find most interesting, or we try to take on a subject that we don't usually take on.
Yeah. Yeah. Accurate. Okay. So like I said, I'm going to do a tiny show and tell that is evolution themed.
It's like we got each other a present. I gave you a little dog story and you're giving me a little evolution story.
Yes. Yes. We're so thoughtful. Okay, so I'm going to tell you about a really interesting new finding that was published in Science Advances, and it is related to lead and human evolution. Oh. So, lead is bad. It is toxic. I feel like we've talked about lead at various points in various episodes, but we should probably, side note, I feel like we should cover it more fully. Like, I feel like we should do a really lead-centric episode. Maybe we talk about ancient Rome. Maybe we talk about, I don't know. There's a lot to talk about.
Yeah.
Regardless, lead is often thought of as something that is fully related to or result of humans producing things like paint and, leaded gasoline and using it for things like mining. But now researchers have analyzed 51 fossil teeth from a range of hominids and great apes. So we're talking like Australopithecus. We're also talking Neanderthals and then Homo sapiens. And what they found was lead. Lead in their teeth, showing exposures going back nearly 2 million years. Wow. This is where we get into the millions. So like it could have come from soil or volcanic activity, for example. And so why do we care? Why does that matter? They're all dead. But what it means is that our ancestors' brains were developing in the presence of what we now know is a toxic metal.
Right.
So what was the impact of that? So to try and figure that out, they did a number of things, but this is the part of the research that I think is really interesting, is that they used brain organoids, which are often referred to as mini brains. So these are like super simple models of early brain development. Essentially what you have are different cell types that are like trying to recapitulate the different layers in the brain. And so you are able to learn a lot from brain organoids at this point.
Yeah.
And so what they did was they tested how lead affected two versions of a gene that is important for neurodevelopment. So one version was the one you would find in modern humans. The other is the one that you would find in Neanderthals and other extinct hominids. And what they saw was that with lead exposure, the Neanderthal variant fared a lot worse. They saw issues in neurons and brain regions, particularly those that are important for language and speech development, which I think is fascinating. Yeah. Fascinating. And so, like the ability for our most closely related ancient ancestors to withstand lead exposure could have driven genetic changes that allowed us to survive and also influenced our ability to communicate and use language.
Oh my God.
Yeah. I mean, this is, again, this is 51 fossil teeth. They're using organoids. It's so hard with things that are this ancient. It's so hard. But I think that it feels like this theory has legs.
Right.
It's really so interesting.
That's so cool. Yeah, I just when you were saying that, I was thinking about it because my son has just started really, his language has been really expanding. Like over the last few weeks, his vocabulary has really been expanding. Yeah. And so it's like that excitement of watching someone assign words to things. And I mean, that's one of the cool things. I assume you're experiencing this too, of watching a baby where you're oh, right, this is how humans become human. They go through this process of physically learning how to do things, and then mentally, they're putting together things with communication and everything. And it's just so cool to like see potential mechanisms for like how we've gotten to be this way.
Yeah.
But also like it's so weird to be like, lead is, I mean, it's still bad for us, but it wasn't that bad for us, I guess.
Yeah,
but it's still bad. Don't eat lead.
It's still really, really, really bad, especially for kids. It is very, very bad. Definitely don't want to downplay how horrible lead was, but it appears it was even more horrible for our ancestors, or at least to the point where, or at least it's proposed that it influenced their development. Really hard to test that on a Neanderthal because, hey, they're not here anymore. They're gone. So yeah, I just thought that this was so fascinating. And I thought it was really cool how they combined, fossil analysis with, some like biochemical analysis plus organoids.
For sure.
Plus, genetics. It's so cool.
Yeah.
We love evolution today.
We sure do.
And every day. We always love evolution.
Glad it got us here. Thanks for tuning into this week's episode of Tiny Matters, a podcast brought to you by the American Chemical Society and produced by Multitude. This week's script was written by me and edited by Michael David and Sam, who is also our executive producer. It was fact-checked by Michelle Boucher. Our audio engineer is Misha Stanton. The Tiny Matters theme is by Michael Simonelli and the Charts and Leisure team.
Thanks so much to Anne Tarrant and Hua Lu for joining us. Go rate and review us wherever you listen. We super duper appreciate it. We are coming up on 2 weeks from now, episode 100. We are closing out the year with episode 100. So let's close it out strong. Please leave us a rating, a review. It's really, really important to us, to the people who listen to our podcast, to the people that make our podcast possible, that we get those ratings and reviews. So please help us.
Yeah.
Give us these are our Christmas gifts,
yes.
New Year's gifts, whatever it is, whatever holiday you celebrate, that gift.
Yeah.
And as always, if you have an episode idea, email us tinymatters@acs.org. And we'll see you next time for episode 100.
That was really bad dramatic music.
No, I loved it. Keep it, Misha. Keep it.